Good day and welcome to our last part of our streptococcal infection lecture series. And in this part 4, we are going to discuss mainly endocarditis, infective endocarditis caused by streptococca. So that's what we call streptococcal infective endocarditis. I am Dr. Teke Apalata, a clinical microbiologist. So let me share the screen with you on this last part of our lecture on streptococcal infection. We will mainly focus on infective endocarditis. So if you missed our previous 3 lectures, please go and review them. So what are the streptococcal species that are involved in causing infective endocarditis? It's mainly what we call the oral streptococca, and they were initially called the viridans streptococca or the viridans group, but we call them oral streptococca. They involve like Streptococcus salivarius, sanguis, mutans, and mitis. And we also have the Enterococca, like your Enterococca faecalis and faecium. So those are commonly involved in causing infective endocarditis. So oral streptococca and enterococca are highly involved in the development of infective endocarditis. So I said that they were formerly called the viridans streptococca, and they form a mixed group of microorganisms with different species. And what is particular about oral streptococca is that those microorganisms of this group are almost always susceptible to penicillin G. And that is a good thing. They are always almost susceptible to penicillin G. But there is another subset of oral streptococca that we call the miliary group, Streptococca miliary group, or Streptococca aeruginosus group. So they involve Streptococca aeruginosus, intermedius, and Streptococca constellatus. Those should be distinguished from this first group because most of these are mainly involved in causing mainly deep-seated abscesses that will then disseminate hematogenously, and it might also lead to the development of infective endocarditis, but those are more common than this group. Now, the reason those should be differentiated from that, it's because of the management purposes, because often those will require a long duration of antibiotic therapy, and you need also to try and find out if there is an abscess somewhere that needs to be drained. Okay. Then there is also another subset of the same group that we call nutritionally variant defective streptococca that have been recently reclassified into other species. Some are called Ebiotrophia and Granulicatella group. So it's also very important to exclude this group because from a management perspective, since those groups are often tolerant to penicillin, and it means that the minimal bactericidal concentration is much more higher than the minimal inhibitory concentration. So it makes them to become tolerant to penicillin. Then we have the group D, other group D streptococca that are non-enterococca. Group D, like your Streptococca bovis, those are like oral streptococca. They are usually sensitive to penicillin, And the problem with this, if you find a patient with these group D streptococca or Streptococca bovis, you need to exclude the possibility of a tumor in the bowel. So it needs to be explored. And among the Enterococca group, mainly your Enterococca faecalis and Fishium, and to a lesser extent, Enterococca durans. Those are also the 3 species that are commonly associated with infective endocarditis, like your oral Streptococcal group. Now, what happens before developing, so that the patient develop infective endocarditis? So the first thing is that the patient should have a pre-existing valve endothelium damage, in the heart. There should be some kind of damage in the heart. So this happens in many ways. It can be provoked by turbulent blood flow that can damage something in the valve. It can be the use of electrode, or catheter, in the valve or in the heart, and inflammation, patient who develop rheumatic carditis, or maybe it can be as a result of some degenerative changes in very old patients, individuals, as a result of either inflammation or micro ulcers or microthrombi. So there should be some kind of valve endothelium damage, and then this will then lead to a local inflammation. So the local inflammation will then trigger what we call endothelial cells to express integrins. Those integrins belong to the beta-1 family, and they bind with the circulating fibronectins to the endothelial surface. Now, pathogens that are able to cause infective endocarditis, like your oral Streptococca and the Enterococca, they carry what we call the fibronectin binding proteins. And because they carry the fibronectin binding protein, they are then able to target, the fibronectin that is attached to the endothelial, to the valve, that are attached to the valve, because we say that the endothelium cells that are found there, they express integrin, and those integrin, they are able to bind the circulating fibronectin. Then those microorganisms during a transient bacteremia, because they carry fibronectin binding protein, they will then attach to those fibronectin. Then they stay there, they replicate, and they develop infective endocarditis situation. So we say that you need to have some pre-existing endothelium valve damage. Then there is a condition that can cause a transient bacteremia, so the presence of bacteria into the bloodstream. And some of the procedures, invasive procedures, might happen, like a tooth extraction that can lead to some of the oral bacteria or oral Streptococca going through the bloodstream. And if they found some kind of valve damage into the heart, they can then attach there. Or other simple procedure like tooth brushing or chewing, it can release some of the microorganisms to go into the bloodstream. So there is that spontaneous bacteremia that can also happen, with some few bacteria moving into the bloodstream. They can explain some of the cases of infective endocarditis that we have that are not related to any invasive procedure. Okay, so the classical infective endocarditis pathogens share the ability to adhere to those damaged valves, as I said, and they trigger local procoagulation activity and then nurture infected vegetations in which they can survive. So they then form, there is a procoagulation activity, they then form in the heart what we call vegetation. And this is an environment that allows them to survive, to continue to replicate inside the vegetation. So they are equipped with numerous surface determinants that mediate adherence to host matrix molecule present on the damaged valve, like fibrogen, fibronectin, platelet proteins. So they then trigger platelet activation, and you will see that the valve will keep growing. So following colonization, adherent bacteria escape the host defense. And one of the ways it's allowed them to escape the host defense is because they have what we call PMPs. So they may be the target of platelets. There is what we call platelet microbiocidal proteins. So platelet microbiocidal protein are released by the platelet, and those infective endocarditis causing pathogens are able to escape the effect of the PMPs. So they are the ones that are able to escape the effect of the platelet microbiocidal proteins. So it is one of the characteristics. Now, how do we make the diagnosis of infective endocarditis? We use what we call the modified Duke's criteria. The modified Duke's criteria has major criteria, and we have two big major criteria. One is related to blood culture that should be positive for infective endocarditis. And the second one should be imaging techniques that should be positive for infective endocarditis. And we have 5 minor criteria. So the first major criteria, we say you need to have blood culture positive to confirm infective endocarditis, and you should find typical microorganisms that are consistent of causing infective endocarditis from at least 2 separate blood cultures. And here I speak about 2 separate sets, blood culture sets. One set is 2 bottles. So 2 separate sets, take at different intervals. And they all come positive either with your viridans or oral streptococca, or the Teke group, but here we are mainly focusing on streptococcal infection. Or we can have microorganisms that are consistent with infective endocarditis from persistently positive blood culture results. So you have 2 or more positive blood cultures of blood samples drawn more than 12 hours apart. They can also give you an indication, or all of 3 or a majority or more than 4 separate culture or blood, they give you the same thing, consistently the same thing. So they are consistently, those blood culture are consistently positive. So what I'm saying is, if you suspect infective endocarditis, you collect blood culture, and you need to collect at least 3 sets. You need to collect a 3 sets maximum. There you can at least 2, but maximum 3 or even 4 different sets of blood culture taken at different intervals. Then it either you grow microorganisms that are known to classically cause infective endocarditis, like the oral streptococca or enterococca, or you can grow another pathogen but consistently found still in the blood culture result when you take 3 or 4 different sets of blood culture. Now you can also do imaging techniques like echocardiogram. Echocardiogram can be positive. You can see the presence of vegetations. When you do echocardiogram, you can find vegetations. You can find abscess or pseudoaneurysm. You can find valvular perforations, or you can find abnormal activities around the site of prosthetic valve implantation. So there are those typical abnormalities that you can find by doing echocardiogram that you can be able to detect. So those are the 2 major criteria. The other minor criteria is like predisposition, such as any existing heart condition, or someone use drugs, injection drug users, or you have fever, you have vascular problems, you have immunological problems, or microbial evidence like positive blood culture, but it does not meet the major criteria that we explained. So those are the 5 minor criteria that you can have. Okay, so you definitely make the diagnosis of infective endocarditis if you have a combination of those pathological criteria and clinical criteria. So pathological criteria, you have microorganisms that are demonstrated by culture, or you have pathological lesions, like vegetation that you find in echocardiogram. So if you have two major criteria only, those two major criteria, you can make the diagnosis. Or if you have one major criteria plus three minor criteria, you can make the diagnosis. Or if you have all five minor criteria, you can also make the diagnosis of infective endocarditis. So that is definitely, you confirm the diagnosis of infective endocarditis. But a possible infective endocarditis is when you only have one major and one minor criteria, or you have only three minor criteria. There is a possibility of an infective endocarditis, but it's not final. Otherwise, the diagnosis of infective endocarditis could not be confirmed. Now, how do we manage this? We manage this, I say that if you have strands of Streptococca that are susceptible to penicillin, they do antimicrobial susceptibility testing in the lab. They say that they are susceptible to penicillin with a MIC, minimum inhibitory concentration of equal to or below 0.125 milligram per liter of those oral Streptococca, then you can treat this patient with penicillin G or amoxicillin or ceftriaxone for a maximum of 4 weeks. And the doses are there, so you can treat for a maximum of 4 weeks. But you can also shorten the treatment in 2 weeks by using that antibiotic, combining it with an aminoglycoside, mainly gentamicin. So then you can shorten your treatment for 2 weeks if you combine penicillin and gentamicin, 4 weeks if you use penicillin alone. In patients who are allergic to beta-lactam, you can use vancomycin still for 4 weeks of treatment. But if the lab isolates strands of Streptococca that are resistant to penicillin with a MIC, it's between 0.250 to 2 milligram per liter. So it's a very high MIC. Then what you do is you increase the dose. Remember here, the dose of penicillin were between 12 to 18 million units per day. But here you use up to 24 million units per day. So you increase the dose of penicillin or amoxicillin, then you combine it with gentamicin, and you also extend the duration of treatment to 4 weeks. But the duration of aminoglycoside should not go above 2 weeks because of toxicity reasons. So you keep the gentamicin for 2 weeks, but you use penicillin high dose up to 4 weeks. And the same in patients who are allergic to penicillin, you can use vancomycin for 4 weeks, but the dose of vancomycin is still 30 milligram per kg per day, because of the toxicity also associated with vancomycin. But here, you use it for 4 weeks, you combine it with gentamicin for 2 weeks. So this is infective endocarditis caused by streptococcal infections. It can be oral streptococca or enterococca that are the commonest, that are known to be the commonest causes of infective endocarditis. And this was the last part of our lecture series on streptococcal infections. So it was part 4. If you missed the previous 3 parts, you can go and review them, where we discussed about different infections that are caused by streptococca. Thank you and goodbye.